Hello dear students, we are starting with a separate section of microbiology from this session, that is parasitology. So we have seen various bacteria, fungi which causes infections. So this section is dealing with the parasites which causes various human infections. So how will you define a parasite? A parasite can be defined as any organism which depends on a living host for its requirements, for its survival. It derives nutrition from that host without giving any benefit to the host. So it is not a mutual symbiosis kind of relationship. Here, only the parasite is deriving benefits from the host, and the host in turn may be harmed by the parasite. So that is what is a parasite, defined as an organism which depends on a living host for its survival and derives nutrition without causing any benefit to the host. So a parasite, it needs a host for survival, and some parasites may need more than one host, or it can be two hosts or three hosts depending upon the type of parasite. So a host can be defined as, again this definition also needs to be noted, can be defined as an organism which harbors the parasite or which provides home for the parasite. So the parasite will be developing inside this host. It can have different types of developmental cycles, like certain parasites divide by sexual means or asexual means or both. So it is the host in which the parasite divides sexually or replicates, reproduces sexually is called a definitive host. So the parasite will complete its life cycle in different hosts, which is the host in which it divides or multiplies sexually. You call that host as a definitive host. Now, the host in which the parasite divides or reproduces asexually is called an intermediate host. So we have a definitive host and an intermediate host for most of the parasites. In some parasites, both the definitive and intermediate will be the same host. Everything will be taking place in the same host. So that is a single host parasites. So, but most of the times, but normally we'll see definitive host maybe one like man, intermediate host maybe some other organism, it can be an insect like a mosquito. So the best example that you can learn for this, when this is asked as a short note, define host with examples, you should say host is an organism or a living thing which harbors the parasite, in which the parasite divides or reproduces. So, for example, the malarial parasite, you call the malarial parasite as Plasmodium. Plasmodium is the malarial parasite. So, Plasmodium has got two hosts, one definitive host and one intermediate host. So, the definitive host of Plasmodium is actually a mosquito which belongs to the Anopheles mosquito. So, it is a female Anopheles mosquito that is the definitive host for Plasmodium, in which it reproduces sexually. The sexual reproduction of Plasmodium takes place in a mosquito. Whereas intermediate host is man, in which the parasite undergoes asexual reproduction. The same thing if you take the filarial worm or the filarial parasite, which is called as Wuchereria. Wuchereria is the filarial parasite, in which also you have a definitive host and intermediate host. Here the definitive host is man, where the worm reproduces sexually. An intermediate host is actually mosquito belonging to a species called Culex mosquito. So like this, different parasites will be having different definitive host and intermediate host. You can see in some parasites, man acts as a definitive host. For some others, it acts as an intermediate host. And for certain parasites, like certain worms, like roundworm, hookworm, those kind of worms, man is the only host. Absolute life cycle, the entire life cycle takes place in a single host. Similarly, we have another kind of host called the reservoir host. You know what is reservoir host? Reservoir host is that living thing in which the parasite lives for a long time, but it won't divide sexually or asexually. This just provides a home to the parasite where it can act as a reservoir for the parasite. So that is what is called as a reservoir host. Now we are going to see the classification of parasites. You know, classification of parasite is very important because it can be asked as a question for exam. So you have basically two groups of parasite. You classify them broadly as protozoa and helminth. Protozoa and helminth. So actually protozoa are unicellular parasites. Unicellular parasites and helminths are multicellular parasites. So you have unicellular parasites and multicellular parasites. Unicellular parasites is called as protozoa and multicellular parasites are actually, you can call them as worms. So they are called helminths. So coming to the unicellular parasites first, that is protozoa, you have to memorize which are the parasites that are coming under protozoa. The most common one which you all may be familiar with is amoeba, and the amoeba species that infects man and is pathogenic to man, the most important species is Entamoeba histolytica. Entamoeba histolytica, which causes intestinal and extra-intestinal amoebiasis. That is what we are going to learn in detail in this session. Other than Entamoeba histolytica, there are also free-living amoeba which can live outside a host. It can live freely in the soil and water and all. But at times when it enters humans, they can cause infection like encephalitis, keratitis and all. So the free-living amoeba examples are Acanthamoeba and Naegleria. So these two names you have to learn. You don't have to learn in detail about them, but just memorize the names because suppose a question comes, name two free living amoeba, you have to write the names Acanthamoeba and Naegleria. Coming to the next type of protozoan ones, this amoeba, it is unicellular and it moves by forming extension of its own membrane that is called pseudopodia. So how does amoeba moves? Amoeba moves by pseudopodia. This you might have learned in your school classes. So that is the motility factor for amoeba. But you have other protozoa which have flagella arising from them so that they can move with the help of flagella. So flagellated protozoans are called as flagellates. Flagellated protozoans, you call them in general as flagellates. And the best examples for flagellates are Giardia lamblia, which causes intestinal giardiasis. It's an intestinal parasite. Then you have Trichomonas vaginalis, which causes vaginal infection, especially in females. Then there are other flagellates. So these two flagellates, you call them as lumen dwelling flagellates. Why? Because they live in the lumen. For example, Giardia lives in the lumen of intestine and Trichomonas lives in the genitourinary tract. So they are called as lumen dwelling flagellates. Whereas there are other flagellates which lives in the blood and causes a disseminated systemic kind of infection. So such flagellates are called as hemoflagellates. Why you call them as hemoflagellates? Because they live in blood. So what are the examples for hemoflagellates? Trypanosoma brucei and Trypanosoma cruzi and also Leishmania. Leishmania donovani causes the famous disease called Kala Azar, and there are also other Leishmania species, whereas Trypanosoma brucei causes a condition called as sleeping sickness. It's called African sleeping sickness. It's because the symptoms in that condition is like that, like drowsiness, confusion, everything will be there in Trypanosoma infection. You call that as a sleeping sickness. And Trypanosoma cruzi causes a condition called as Chagas disease. Just be familiar with these names. Chagas disease is caused by Trypanosoma cruzi and sleeping sickness is caused by Trypanosoma brucei. Sleeping sickness is caused by Trypanosoma brucei. So that is about the Trypanosoma species and Leishmania. Both of them are together called as hemoflagellates and their locomotor organ is the flagella. Coming to the next group that is called sporozoa or they call sporozoan parasites. The best example, the most important example among this group is Plasmodium, which causes the famous malaria. So we all know that malaria is a very important disease of mankind and the causative agent is called as Plasmodium. There are different species of Plasmodium like Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae and Plasmodium ovale. So these are the four species of Plasmodium that causes malaria. All of them come under sporozoan parasites. Then additionally, sporozoan parasite also have an important member called Cryptosporidium parvum. Cryptosporidium parvum is famous because it causes diarrhea in HIV infected individuals. So Cryptosporidium causes diarrhea in HIV. So what is the most common parasite? It's an opportunistic infection in HIV individuals. This kind of organism, Cryptosporidium, generally don't cause infection in immunocompetent individuals. Only in immunocompromised, they cause opportunistic infection and they present as diarrhea. Then other than Cryptosporidium parvum, there is another one called as Toxoplasma gondii, which is also an opportunistic infection. This can cause encephalitis in HIV. Encephalitis in HIV. Now, when we say this, you can also remember two more parasites which causes diarrhea in HIV. Those are Isospora and Cyclospora. So suppose you get a question, name parasites which causes diarrhea in HIV. You can list, one is Cryptosporidium parvum, second one is Isospora, and third one is Cyclospora. All these belongs to sporozoan group of parasites, protozoa. So that is the third group of protozoa called as sporozoa. They have a stage in their life cycle called sporozoid. That is why you can call them as sporozoa. Then finally, the last group of protozoan parasite is called ciliates. Why? Because they have cilia arising from their cell membrane, which helps in motility. So they are ciliated parasites, and the one and only example for a ciliated protozoa is Balantidium coli. Balantidium coli also causes an intestinal disease. It has got cilia as the locomotor organ. So these are the four groups of protozoan parasites. That is, we have seen amoeba, then coming to flagellates, both lumen-dwelling flagellates and hemoflagellates, then sporozoa, and finally ciliates. So please learn examples for each group of protozoan parasites. Coming to the next group, that is the multicellular group, you can commonly call them as worms or helminths. Okay, so they have three important groups: cestodes, trematodes, and nematodes. These are the three classes of helminths. And cestodes are actually flat worms. They are called tapeworms. Cestodes are flat worms. You call them as tapeworms. Why? Because it looks like a tape-like segment with segmentation. It looks like a flat tape. And the common species coming under this class are Tinea solium and Tinea saginata, which can cause intestinal teniasis, and Echinococcus granulosus, which is called as dog tapeworm, which causes a disease condition called hydatid disease. So these are the tapeworms or cestodes, commonly Tinea solium, Tinea saginata, and Echinococcus granulosus. The next group is called trematodes. Trematodes are actually a fleshy kind of worm. It's very fleshy and it's called as flukes. And the best examples for trematodes are Schistosoma hematobium, Schistosoma mansoni, and Schistosoma japonicum. And this Schistosoma hematobium mansoni and japonicum, they are called as blood flukes because their habitat is the venous plexus. It can be different venous plexus, it can be mesenteric venous plexus or vesical plexus. So you call them in general as blood flukes. Now there are other flukes like Fasciola hepatica and Clonorchis sinensis. These are called as liver flukes. So you have to memorize all these examples for blood flukes coming under the genus Schistosoma, Hematobium, Mansoni, and Japonicum. Liver flukes examples are Fasciola hepatica and Clonorchis sinensis. And the last group of helminth and the last group of parasite, that is called nematodes, compared to all other groups, this is a more well-developed form of parasite which has got the well-developed GIT, and it has got separate sexes. Male and female worms are separate. So they are a kind of well-developed parasites. You have different worms under this which are commonly called as roundworm, hookworm, whipworm, and pinworm. These names are given based on their appearance. Roundworm is cylindrical. Hookworm has got hooks at the anterior end, or at the top of near their mouth. They have this hook like attached for attachment. And whipworm looks like a whip, and pinworm is very thin thread-like worm. So roundworm, the correct name for roundworm is Ascaris lumbricoides. That is roundworm. Then hookworm, the correct name is Ankylostoma duodenale. The other names, actually, the other common names that we call roundworm, hookworm. So the classical name is Ankylostoma duodenale. The name indicates the habitat of this worm is duodenum. Then you have the Trichuris trichura, which is the whipworm, and Entrobius vermicularis, which is the pinworm. So these are the nematodes, and all these four nematodes, their habitat is human intestine. So they are called as intestinal nematodes. You have one more intestinal nematode called Strongyloid stercoralis, which causes opportunistic infection in HIV patients. It can cause a generalized strongyloidiasis in HIV patients. So all these five are actually called intestinal nematodes. Ascaris and Ankylostoma, they inhabit the small intestine, whereas Trichuris and Entrobius inhabit the large intestine. And there are other group of nematodes which are seen in the tissues. It can be in the subcutaneous tissue, it can be in the lymphatics. So you have the Wuchereria bancrofti, which causes the famous disease filariasis, and this worm lives in the lymphatics. So this worm lives in the lymphatics. It's called Wuchereria bancrofti. There is one more filarial worm which causes lymphatic filariasis, that is called Brugia malai. Now there are three others, Lova lova, Onchocerca, and Dracunculus, which causes subcutaneous filariasis. So these worms live in the subcutaneous tissue. So these are the classification of parasites broadly into protozoa and helminths. We have seen four classes of protozoa and three classes of helminths. Helminths, cestodes, trematodes, and nematodes. Cestodes are called tapeworms, trematodes are called flukes, and nematodes are actually the cylindrical worms like roundworms. So all these examples you should be thorough with. You have to learn the examples with what is the disease caused by each of them. So we are moving on to entamoeba in detail. That is the first parasite that you have to learn, which is always asked for exam. The morphology of entamoeba, intestinal amoebiasis, extraintestinal amoebiasis, all these are asked in the previous university exams. So entamoeba histolytica, it belongs to the group protozoa because it is a unicellular organism. Habitat of entamoeba is the large intestine of man. See, this lives in the large intestine of man. Entamoeba has got three morphological stages: trophozoite, pre-cystic stage, and cystic stage. So we'll see the morphology in a little bit detail with diagrams because that can be asked for exam. So morphology of amoeba is a previously asked question with figure you have to learn. So this shows the trophozoite form. Trophozoite form is the dividing form of entamoeba, which divides by binary fission. So what happened in trophozoite form, you can see it has got a endoplasm and also an ectoplasm. So this is the endoplasm and this is the ectoplasm. Here you can see the nucleus with the karyosome, and there are different vacuoles inside this trophozoite. They are called the foot vacuoles. So these are actually the foot vacuoles of amoebic trophozoites, to which they derive food nutrition from the host. So it is present, the food that they ingest, it's seen in a vacuole form, so food vacuoles. And there are the ingested RBCs, these red ones are the RBCs. Also there will be ingested WBCs and ingested bacteria. So trophozoite, you can see the ectoplasm has a protrusion at one end. That is how this amoeba moves, and that protrusion is called the pseudopodium. So if this is a trophozoite, it will be having an endoplasm and an ectoplasm. Endoplasm will be having the nucleus, and this is the ectoplasm. Other than the nucleus, what all are there inside the endoplasm? There are food vacuoles, there are RBCs, WBCs, and also ingested bacteria. So this ectoplasm forms protrusions on its membrane for moving the amoeba. For motility of entamoeba, it forms protrusions from one end, and those protrusions are called as pseudopodium. Please don't forget this. So this is how an entamoeba trophozoite looks like. So this is the dividing form of amoeba, and this is the pathogenic form that causes the various clinical manifestations. So then the next stage is the pre-cystic stage. That is when trophozoite is going to get converted to a cyst. In between, it has got a pre-cystic stage, which is intermediate between the trophozoite and cyst. That is, it is going to become a cyst. Encystation has started. Now, what happened when it becomes a cyst? Initially, it will be having this one nucleus of trophozoite will divide to form two nucleus. So you will have a binucleate cyst, and then it will divide to form four nucleus. So nuclear division takes place inside, and thus it forms a quadrinucleate cyst. So here you can see the quadrinucleate cyst, quadrinucleate cyst, and this is the infective form of entamoeba. So we usually get infection by consuming food or water contaminated with the entamoeba quadrinucleate cyst. You can see the four cysts here, and also you can see some chromatic bars. So that is also present in the cyst, and this is the cyst wall, which is a resistant stage. Cyst is actually a resistant stage of the parasite with which it can survive in the adverse environments. And trophozoite form is seen only inside in the human intestine where it is starting dividing. So infective form is a mature quadrinucleate cyst. Mode of infection is ingestion of water and food contaminated with the cyst. So when the cyst reaches the cecum, it is the cecum and the terminal part of the ileum where entamoeba becomes the trophozoite. So it will undergo excistation. Excistation means if this is a cyst with four nucleus, it will undergo excistation, that these four nucleus which is dividing, it will become trophozoites, and finally actually 8 trophozoites are produced from a single cyst. So excistation takes place and it is converted to trophozoites, and trophozoites will divide by binary fission. So we'll see the life cycle of entamoeba histolytica in this figure. Here you can see a person who gets infected by consuming contaminated food or water or through flies or infected fingers. He is ingesting the quadrinucleate cyst, which is the infective form. So what happens to this quadrinucleate cyst is there is a dent in the cyst wall, and the nucleus starts multiplying inside the cyst. It becomes 8 nucleate metacystic stage, and this metacystic stage, it will then get converted into a metacystic trophozoite. So now this is going to give rise to 8 trophozoites from this. So the cyst is getting converted, excisted, and getting converted to trophozoite. And these trophozoites, they will multiply by binary fission, they will move by forming pseudopodia, and they will release certain enzymes which can lead to the damage to the intestinal cells, leading to necrosis and formation of amoebic ulcers. The classical description of an amoebic ulcer is a flask-shaped ulcer, and as a result of this necrosis and inflammation that it produces, it will lead to blood. Uh, it will lead to dysentery, that is, blood will be present in the feces, blood and mucus will be present in the feces, and finally, this trophozoite give rise to a mature cyst. It is going to become a cyst, that is, it is getting encysted. So, trophozoite will lead to initially a pre-cystic stage, which is getting converted to a cyst stage. So it starts from a cyst, it gets converted to a trophozoite. This trophozoite will produce all the pathogenic features like amoebic dysentery, and it form amoebic ulcers. And as a result, after doing all these things, this trophozoite again gets converted to a cyst form. In between, it will form a pre-cystic stage. Finally, this cyst becomes mature into a quadrinucleate cyst, and that is what is excreted in the feces of the infected person. So the life cycle of amoeba is actually simple. It starts from cyst, becomes trophozoite. It causes all the disease condition, that is, trophozoite is responsible for the pathogenicity of entamoeba. It is again converted back to cyst, and this cyst is again excreted in feces. So this is what happened in entamoeba, which is a simple life cycle. So this is what I told. Amoeba produces a proteolytic enzyme that can lead to tissue necrosis. This leads to flask-shaped ulcers, and that is the reason for excreting blood and mucus in the feces, because of the damage it produces. So it leads to amoebic dysentery and also amoebic ulcers. So that is the two main pathogenic feature of intestinal amoebiasis. So you can see blood and mucus is excreted in feces in such a patient. So we'll see some clinical features of intestinal and extraintestinal amoebiasis. You have to know what are the organs which are affected by extraintestinal amoebiasis and how does it occur. Generally, the incubation period of amoebiasis is say 1 to 4 weeks. And what you can remember for intestinal amoebiasis is what I discussed in the last slide. That is, it produces amoebic dysentery and amoebic ulcers. Because of the ulcerative lesions, there is a profuse bloody diarrhea. So the ulcers can be localized to ileocecal region or sigmoidorectal region, or it can be present over the entire colon. It can lead to a entire colitis involving the entire colon. And how will you diagnose intestinal amoebiasis? Your sample will be stool sample, and from stool sample you can look for the presence of cyst and trophozoites in stool sample. And also you can do the blood examination for finding out the antibody serological examination and all. But basically the test for entamoeba, it is mostly confined to stool examination for the cyst and trophozoite, or you can look for amoeba antigens in the stool, or you can do a PCR from the stool sample. That is about the intestinal amoebiasis. So don't forget amoebic dysentery and also the amoebic ulcers that it produces. And what happened in extra-intestinal amoebiasis? How does it occur? Actually, entamoeba lives in the intestine. How it goes to the extra-intestinal sites? This is when this enters the trophozoite from the intestine, enter into the systemic circulation, and it goes to organs like mainly liver. Mainly it goes to liver, and also it can go to lung, brain, spleen, skin, etc. Basically, extra-intestinal amoebiasis is mostly confined to liver, and it causes hepatic amoebiasis or amoebic liver abscess. So that is what you should remember about extra-intestinal amoebiasis, hepatic amoebiasis or amoebic liver abscess. So there is a classical description of the pus that entamoeba produces within the liver abscess. It is called as anchovy sauce pus. It is very brown, reddish brown in color, and this pus is described as anchovy sauce pus. That is a classical description of hepatic amoebiasis or amoebic liver abscess. And how will you diagnose a liver abscess caused by entamoeba? You can do a diagnostic aspiration from the abscess or liver biopsy, and you can look for the presence of trophozoites in the liver abscess, and also you can check for the presence of antigens and antibodies in blood and serum. And a stool examination can also be positive in most of the cases, because this starts as an intestinal amoebiasis and then progresses to extra-intestinal amoebiasis. So don't forget amoebic liver abscess and anchovy sauce pus. So you should know some differences between amoebic and bacillary dysentery. You know, dysentery is produced not only by entamoeba, but it can also be produced by a bacilla that is called as Shigella. Shigella can be different species: Shigella dysentery, Shigella soni, and Shigella flexneri. So how will you differentiate if a patient present with dysentery, whether it is amoebic dysentery or bacillary dysentery? There are certain features. First of all, you can look at the gross observation, macroscopic observation of the diarrhegenic stools. That is, how many times they are having this, how many episodes they are having, how many episodes of diarrhea. It's like if it is 6 to 8 amoebic dysentery, the episodes will be comparatively lesser when compared to bacillary dysentery, which is over 10 times a day. And coming to the volume of stool they passes, it is relatively large in amoebic dysentery and small in bacillary dysentery. Amoebic dysentery, the odor will be offensive smell, and it will be very dark red in color. Whereas in bacillary dysentery, there is no odor, no odorless, and it's very bright red in color. And when you examine the stool, it will be mixed with blood and mucus in case of amoebic dysentery, whereas in bacillary dysentery, they will be just excreting blood and mucus, and there is no feces. Actually, it's mostly blood and mucus that is excreted, not feces. So microscopic, if you examine the microscopy of the stool in amoebic dysentery, you can see there is clumps of RBCs in the stool, whereas in bacillary dysentery, RBC will be in a roller formation and will be bright red in color, and pus cells will be scanty. This is easy to remember. You know, always pus cells are numerous only in case of bacterial infection. In a parasitic infection like protozoan infection, pus cells will be less. And trophozoites of Pentamoeba histolytica you can see in the stool of an amoebic dysentery, but not in bacillary dysentery. And one more thing, for most of the parasitic infections, you can see the Charcot-Leyden crystals present in stool. So that is present in amoebic dysentery. So you can remember at least 4 points regarding amoebic and bacillary dysentery. The easiest points will be pus cells are plenty in bacillary, less in amoebic. Charcot-Leyden crystals are present in amoebic, it is absent in bacillary. Trophozoites of entamoeba present in amoebic, not in bacillary. And what about the number of episodes of diarrhea? Like it will be more in bacillary dysentery. It will be of offensive odor and large volume in case of amoebic dysentery. So like that, it will be dark red in color in amoebic dysentery. All the points which are more easier, you can remember at least 4 points, and that will give you a question like difference between amoebic and bacillary dysentery. You will be able to answer. So the treatment of amoebiasis, if it's an intestinal amoebiasis, the drug of choice is metronidazole or plinidazole. That's what we give for amoebic dysentery. If it's a case of hepatic amoebic liver abscess, you may have to drain the abscess. Surgical correction may be required. And in summary, you can see amoebiasis, it's caused by a quadrinucleate cyst of amoeba, that is the infective form. Mode of infection is ingestion. And what will happen to this cyst when it enters the intestine? It will become a trophozoite. Trophozoite will multiply, it will release enzymes, causes necrosis, and what all things they will produce? They will produce dysentery, that is called amoebic dysentery, and also they will produce amoebic ulcers in the intestine. And what will happen to this trophozoite later? It can get converted back to cyst and it will be excreted in the stool of the same patient. Otherwise, these trophozoites can enter the blood vessels. They can penetrate the blood vessels, enter into circulation, and through circulation, they go to liver or they can go to lungs. In liver, they produces hepatic hepatic amoebic liver abscess, hepatic amoebiasis or liver abscess. And what is a classical description of the pus in amoebic liver abscess? It is anchovy sauce pus. It can be an MCQ question. So these are the points in favor of entamoeba, the best points for entamoeba. So you can get questions like morphology, you have to draw the trophozoite and cyst form. You can also get a question on the pathogenesis of intestinal or extra-intestinal amoebiasis and the lab diagnosis. So when you write lab diagnosis, please write in stool, you look for cyst and trophozoite. So that is about entamoeba. You have the rest of the parasites which we dealt in the next session.